Well, first of all, thank you very much to the organizers for inviting me to come and do this Zoom talk. It's a great pleasure to talk about LRRK2 Parkinson's disease and to talk to you a little bit about where we came from and where we're headed, and the opportunity, I think, to develop meaningful therapeutics to hopefully stop the progression and meaningfully prevent Parkinson's disease in individuals who are LRRK2 heterozygous carriers. So my first slide is just the title and some of the disclosures that I have. These are relevant in some ways because of my former institution patented some of the work I did related to mouse models of LRRK2 biology. I'm going to talk a little bit about them. This is the formula, if you like. I don't know if you can see my mouse, but it's where we're coming from, neurology and neuropathology, really, through population and family genetics, through to modeling in mice for the most part, but I will talk a little bit about some work in C. elegans and cell biology, to opportunities using these insights to develop means higher throughput screens. Higher throughput screens for drug discovery, and some of the work that we've been doing around physiology and imaging, basically with those drugs, to see whether we can rectify the phenotypes that we observe, the detrimental phenotypes observed in LRRK2 Parkinsonism and in these models with various drug compounds to try and put the problem right. I'm going to highlight each of these different areas within the talk, and with LRRK2 G2019S and LRRK2 Parkinsonism in mind. So to start here, this is the beginning. The LRRK2 locus, or Park8 as it was originally called, was mapped in the Sagamihara family in 2002, and this was published by Fanyama Obata in Annals of Neurology. You can see it's a fairly large pedigree, a fairly large kindred with affected individuals labeled in black here from a family, large family. Each symbol labeled in black has Parkinson's disease, and they carry a distinctive haplotype shown underneath, which basically we later learned harbors an I2020T mutation within exon 41 of leucine-rich repeat kinase 2. Fanyama and colleagues published this discovery. The Sagamihara kindred is actually from the Sagamihara prefecture in Japan, just outside of Tokyo. It's a very large family. Subsequently, the 6 members of this family were kind enough to donate their brains to autopsy, and all 6 members of the family had tauopathy. So they had a neurofibrillary tangles in the brain. They had clinically definite Parkinson's disease, but neurofibrillary tangles in the brain in all 6 cases, rather than Lewy body disease, which is the hallmark of Parkinson's disease, pathologically speaking. We followed up on the discovery that year, and 2 years later published our findings from a couple of different families. One of them is shown here, known as Family D, the Western Nebraska kindred. It actually comes from the UK, is its original origin, but you can see again, it's a dominant pattern of inheritance. All the black symbols here are individuals within the family who had Parkinson's disease, and you can see again the pedigree links to chromosome 12q12. 4 members of this family gave their brains to autopsy, and you can see different slides here, different pictures of the pathology. And the fundamentally interesting thing was that we found that different members of the family, although they actually had the same mutation, it was an R1441C mutation, actually had pleomorphic pathology. So some members of the family had Lewy body disease, typical for midbrain Lewy body disease and Parkinson's. Another member of the family had much more widespread pathology and had a pathologic diagnosis of dementia, of diffuse Lewy body disease, dementia with Lewy bodies. And then 2 other family members had only, one family member had nigroneuronal loss and just scoliosis and no inclusion body pathology. And the last family member had a 4 repeat tauopathy and neurofibrillary tangles again. So the pathology in the family is very different, although the clinical presentation was the same. The pathologic presentation was quite different. We went on in 2003, 2004, and working with a good friend of mine, Jan Asli in Norway, shown here. We were working with a series of families in the central mid kind of area of Norway around Trondheim. Trondheim is shown here on the map. And these families come from this region. And what we noted was these pedigrees all had an autosomal dominant pattern of inheritance, so that 50% of each generation is affected, and down the family lines, men and women equally. And what we found is that these families actually had a G2019S mutation, a glycine 2019S change, glycine to serine change, within the kinase domain of the leucine-rich repeat kinase 2 gene. It was curious at the time because we actually found that while all of these Norwegian pedigrees, and they're shown here in orange rather, each of these bars represents a proband from one of the families. They all have a common haplotype. This is the region of chromosome 12q12 inherited identical by descent within each family. That's identical across all of these pedigrees. But we also found the same haplotype in families from the US and from Poland, and further afield as well. And we'll get to that in a minute, mapping down into the southern Spain and across the Med into North Africa and indeed into Tunis. We'll talk about that in a sec. So it was rather surprising for us, this mutation. Not only was it affecting the kinase domain, and we predicted at the time the mutation basically keeps this ribbon of protein here active or open and allows substrate to come in. This is the active site. This is the ATP binding domain, and this is the activation segment, as it's known, of the kinase, which basically normally protects this pocket and prevents aberrant phosphorylation of substrate. Just from the discovery of the protein of LRRK2 itself, we realized that it was very likely that G2019S induced constitutive kinase activity, overactivity in the protein, and that was subsequently shown a few years later. What was also however, there was also many other variants within the protein. So other than G2019S and R1441C, as I've mentioned, and I2020T, there was this most very common mutation, G2385R, which actually turned up to be a risk factor for Parkinson's disease. It's not as pathologic. It doesn't necessarily, it's associated with the disease, and it merely doubles your risk of getting Parkinson's disease. But this particular variant in the WD40 domain, the end of the protein, basically is found in somewhere between 6 and 8% of all patients with Parkinson's disease in Asia. And again, we found that G2385R was related to a common ancestral founder. Here's the variant here, and this segment of chromosome 12q12 basically segregates within everybody who has this mutation within that region. And I'm talking all the way down through from Korea through Japan and through the whole of China down into Taiwan. So it's an awful lot of patients with LRRK2 Parkinsonism, or risk factors for LRRK2 Parkinsonism. A little later on, we explored a lot of variability within the protein. We looked at 120 actually coding changes within the protein in around 15,000 individuals, and found that that there were many other risk factors, and this is just showing a protective haplotype for LRRK2. So there's variability within LRRK2 that promotes risk, and there's also variability in LRRK2 that also is neuroprotective for disease as well. The real insight, however, came and surprise, I think, came as we tracked this mutation down from Norway into Spain and into the Mediterranean. This was done by a number of groups, but my group were very much involved as well. And what we found, and this is highlighted by a couple of papers here in New England Journal in 2006, one from Laurie Zelius's group and one from Suzanne Lesage, is that the G2019S mutation itself actually turned out to be the most frequent. It's found that a frequency worldwide at about I forget exactly, but it's a few individuals in every 10,000. That's the frequency. It's something like 10 to the -4 is the frequency of LRRK2 G2019S. But as you go south from northern Europe into the Mediterranean and into North Africa and across here into Israel, you find that the frequency of LRRK2 G2019S increases. The background frequency of G2019S in the Berber people, the majority of the population in North Africa here, is actually around 1.9%. So it's almost 2%, or 1 in 50 people have the LRRK2 G2019S variants in Berbers. And you find the same background frequency of G2019S in Ashkenazi Jews in Israel as well. One of our thoughts at the time is that the reason this variant, when we discovered the variant in Norway, the reason it got to Norway is probably with the Phoenician trading routes, which are shown here. And there's a picture of Queen Dido. And anyway, this is all population history and speculation, but it's curious as to how this mutation seems to have spread. The largest haplotype and the oldest haplotype for G2019S is in Berbers, and it comes from North Africa. And so we think the mutation actually originated there around 1000 BC. Around this time, around 2004, 2005, I was fortunate enough to meet Faisal Hentati at the American Academy of Neurology, and I came back with him to visit the National Institute of Neurology in Tunis. And this is the picture with Faisal and a colleague here from GlaxoSmithKline. Thinking at the time, if an overactive kinase basically causes Parkinson's disease or is a major risk factor for it, then surely there must be a way of developing competitive kinase inhibitors as a means to slow or even prevent this disease in individuals that are at higher risk for developing it. One of the things we wondered is why the frequency is so much greater in North Africa. Why has this variant that originally occurred once, one event, has increased in frequency to about almost 1 in 50 people within the population? And one of the reasons for this is, and you can see it, if you look genetically at the chromosome 12 region, you can see that there's evidence here, for example, of a positive, this is just shown in a genetic sense as integrated haplotype scores, but you can see a massive positive selective sweep for maintaining LRRK2 G2019S within the population in North Africa. The reason for this, of course, is lost in history and lost in time. But one of the things that has come through from the literature is that a number of genome-wide association studies have now been done for a variety of different chronic kind of inflammatory disorders, including Crohn's disease here, for example, IBD. And they find, these GWAS studies have found very significant risk factors in and around LRRK2, in and around LRRK2, not G2019S itself, but in and around LRRK2, that basically promote the association with inflammatory disorders. So LRRK has a major role in inflammation. This has been shown in a variety of studies since. So, for example, this is a study of some mice, of LRRK2 G2019S mice, heterozygous mice and wild-type mice. Here, the littermate shown here. And you can see this is actually infecting the mice with the salmonella and showing that the wild-type mice with a lot of salmonella, I'm trying to find my mouse, a lot of salmonella infection basically become moribund and die relatively quickly, or within about 10 days post-infection, whereas those with a G2019S mutation, or even homozygous for the G2019S mutation, are far less likely to die or die as quickly because of this genetic advantage that they have, which protects them from this inflammation and this pathogen. However, the double-edged sword is, of course, that it promotes those individuals who have this mutation perhaps the mice, to Parkinsonism-like phenotypes later in life. Looking at the penetrance, and I don't want to be genetically deterministic about this, the penetrance of LRRK2G2019S is far from complete. Just because an individual has the variant, has this particular mutation, it doesn't mean necessarily they will get Parkinson's disease. It just elevates their risk, and this is an age-associated phenomenon. And you can see it here. This is a study we did with Dr. Hentati and with Jan Asley, whose picture I showed you holding the fish. Basically comparing the penetrance, that's the probability of getting the phenotype, if you have that genotype, in Norway versus Tunisia. And you can see here, and we also compared the penetrance in Ashkenazi Jews from Israel as well. You can see here that if you're a Norwegian, have a Norwegian genetic background, your probability of becoming affected is going to be later, and you're less likely, in fact, to develop the disease than if you're a G2019S carrier of Ashkenazi Jewish or Berber genetic background. You can see this major shift in the curve. So the penetrance is not only not complete in all populations, there's also major penetrance differences across different populations as well. Clinically, though, the clinical phenotype is very much the same as idiopathic Parkinson's disease. This is a study that we did with Faisal, looking at around 220 patients with LRRK2G2019S, and around comparing those clinical findings with around 350 patients with idiopathic Parkinson's disease of unknown etiology. They were genetically screened for all the known genetic risk factors at the time, and they didn't have any. And so we compared these groups. And what you can see here is some interesting findings. For example, in these Kaplan-Meier plots here, we find that women generally, just by a few years, if they're G2019S carriers, may manifest Parkinson's disease just slightly younger than males. We also find something very curious in terms of age of onset, and that's that in most patients with idiopathic Parkinson's disease, age of onset correlates in some ways with the severity of the disease. So the longer you've had the disease, the more likely it is that you have a variety of bigger constellation of these features, and they may be more problematic. But that doesn't seem to be the case in LRRK2G2019S. We can show that here in these graphs. We basically find that individuals with later onset disease, the later you are, the older your age when you develop Parkinson's disease, typically the faster your progression. And similarly, or conversely, I should say, the younger you are at age of onset, the more mild and slowly progressive your disease tends to be. And that separates out the curves as mild, moderate, and severe forms of Parkinson's disease by age of onset. This is for idiopathic PD, but this is for LRRK2G2019S. So it doesn't seem to matter when your age of onset, your disease seems to be much more uniform. Nevertheless, we were thinking, we started doing some imaging studies back around 2005, and a number of different studies now have been done looking at by using PETs, positron emission tomography, and 18-fluorodopa scanning, as well as looking at a variety of other markers. So for DAT, for VMAT2, looking at 18-fluorodopa turnover, and this particular figure here is showing DAS binding for serotonin, for the serotonin receptor. We find that although individuals may be clinically, when they're affected, they look like idiopathic Parkinson's disease, and even when they're not clinically affected, they have interesting, quite profound differences in their dopaminergic system. So our youngest patient, youngest individual who was scanned, I think was around 22 years of age, and he's a very active young man and has no clinical signs or symptoms of Parkinson's disease. But in terms of his 18-fluorodopa turnover, it's much more elevated than you would expect for an age-matched control subject of. So you can basically see And this particular figure here shows an increase in serotonergic DASP transporter binding in the striatum long before individuals become patients, suggesting there may be some serotonergic compensation going on, and these individuals masking symptoms before they become patients. Be careful with that. There were some modifiers, and we started doing some studies of LOCT2 modifiers. This is basically trying to explain this phenomena here. For example, in this family, there are 2 individuals who developed age onset Parkinson's disease, 2 brothers at around, at around 40 years of age. However, their parents are both carriers of LOCT2 G2019S. This is a family from Tunisia. The parents are 78 years of age and 72 years of age. They're both carriers. Neither of them have clinical Parkinsonism. They probably have dopaminergic deficits by PET, perhaps, if we did PET imaging. But clinically, motorically, they're perfectly normal. But both of their sons were 40 when they developed symptoms and were later diagnosed. With these types of families, we did another linkage-based study, long story short, and identified a modifier, genetic variants within a gene called dynamin-3, which was the first genetic modifier for LOCT2 G2019S. In the individuals who have a particular genetic variability in dynamin-3, GG homozygous variability here, may manifest disease symptoms if they're LOCT2 G2019S carriers younger than the CC or CG individuals with these particular dynamin-3 variants. In terms of LOCT2 functioning, move on to this pretty fast. I think you go through this. We knew early on that it's a ROCO protein. So by a ROCO protein, it has a particular structure. It basically has a RAS GTPase domain followed by a C-terminal of ROC. It's known as a core domain, and followed on by a kinase domain. It's a particular motif of family, a particular family member, particular family of proteins. ROCO proteins are probably most abundant in Dictyostelium, which is a slime mold, and they've been known to be involved in the remodeling of cytoskeletal machinery, actin-based machinery, and tubulin-based machinery. So they've got an intimate role in reorganizing slime molds. Slime, that's what they do. They reorganize the actin machinery at one end of the cell and promote the actin polymerization at the forward end of the cell to slime across the dish. The earliest work that I'm familiar with, this work in C. elegans here, showing that the LRK1 knockout, it's the homolog in a worm of LRK2, but if you knock it out, basically you lose the polarity of sorting. Here they used a synaptic vesicle protein, synaptobrevin, labeled it up with green fluorescent protein. And normally in a wild type neuron, you see this is preferentially localized to the axon and axon terminals here. When you knock out LRK, you basically get mislocalization of this within dendrites as well as in the axon. So the sorting goes awry. So it's also involved in cargo sorting and vesicular transport of synaptic proteins within worms. Although this is based, obviously there's some caveats here. It's an LRK1 knockout. It's not the LRK2 that we find in humans. But a number of different studies basically followed along, whether it's unbiased proteomic screens showing interaction with a whole variety of actin-based cytoskeletal components. It also showed a role, something that's very interesting about LRK. It's got these, I think, 7 domains, which are this consensus sequence here, which basically target LRK2 itself to lysosomes. So it clearly has a role in autophagy. The protein has a role in autophagy because it's the protein itself is driven there by having these specific motifs. There's a number of studies that have come out over the years, and I'm just doing this historically in a way, including the phosphorylate LRK2 is known to phosphorylate endophyllin which is a protein involved in endocytosis. It also phosphorylates auxilin, which is involved in mediating endocytosis as well. And clearly, the relationship here is that LRRK's intimately involved in regulating synaptic function. Something early that came out in 2012 that I was scratching my head around was if you overexpress these mutant proteins, and just in cells, you overexpress wild-type LRRK2 compared to R1441C or I2020T, this is a Gamma-Hara family mutation, you basically get polymerization of the LRRK, overexpressed LRRK, on microtubules, and you get these skein-like formations formed within these cells. And at the time, I found it very difficult to make sense of this, because you can see they all behave similarly in terms of increasing the binding. The mutant LRRK2 increases binding to microtubules to form these structures, except for G2019S. It's the only odd one out. Very odd to me. And I was wondering, is this just an artifact of overexpression? And for the longest time, trying to figure this out. Of course, we made mice. As I pointed out in the schema in the beginning, you basically look at the pathology, look at the clinical phenotype, map the gene, and basically develop mouse models. And we've made knockouts and we've made knock-ins. We try and stay as physiologic as possible by doing the most subtle alterations in the murine genome. And the knockout mice, Jia Shen's group, Tong et al. Actually beat us to this one, but we replicated their findings. You see an interesting, you don't see any really profound neurologic phenotypes. There's increased stigmataxis in the mice, but you do see this enlargement of kidneys as the mice age. So these are the larger kidneys in the knockouts. They also become discolored, much darker and pigmented. And I think that's the lipofuscin that kind of builds up within them. And you can see that here with autofluorescence. You can also see this, the pigment here that's building up within the kidney tubules. So they tend to have a kidney phenotype if you knock out LRRK2 altogether. But in terms of their neurologic function, they seem to be quite well preserved. In the G2019S knock-in mice, again, the phenotypes are relatively subtle. There's some slight dopaminergic differences, but it doesn't seem to have any profound impact on motoric phenotypes. There is some increase in tau phosphorylation. So microtubule associated protein tau tends to increase in terms of phosphorylation in the heterozygous and homozygous animals, but it's rather mild. And there is increases in aberrant mitochondrial structures as well with old age in those animals. We looked at this to death in a way. We did a really big comparative study of knock-in and knockout mice, and we looked at the electrophysiology of these animals. And what we find is an increased probability of release, which is shown here, in terms of glutamatergic innervation in the striatum. And you can see that there's an increased probability of release in the G2019S animals. And just move this out of the way. You can see that over time, if you compare 3-month-old mice, wild type, and G2019S mice, there's this increased probability of release. You can compare the wild type mice at 3 months versus 18 months, and there's not so much of a difference. If you compare the G2019S mice at 3 months versus 18 months, there's a much more profound difference. It's almost as like there's accelerated synaptic senescence in the animals, and the candle is burning brighter, but perhaps not as long. It's putting a challenge on synaptic transmission that is causing those synapses perhaps to fail with aging. It's one of the hypotheses that we had. The big findings started coming out really, and insights coming out about 2016 with this paper showing that LRRK2 phosphorylates RABs, a whole bunch of RABs, actually 14 different RAB proteins. So RABs are basically the address labels, if you like, for vesicular components. And there are many different RABs that are phosphorylated by LRRK2. You can see a whole list of them here. And that's what the G2019S mutation seems to be doing, is differentially phosphorylating a whole bunch of RABs, which are controlling the trafficking of vesicular components of endosome within cells. Number of kinase inhibitors have been developed. There's a variety here. Most of the companies, so whether it's Pfizer, Merck, and Genentech, have developed a whole variety of different molecules that can basically interact with the kinase domain and silence or inactivate or inhibit rather the kinase activation and do it quite efficiently. But some challenges remain. Oh, some more opportunities first. There's also the RAB-GTPase domain, which is another area of the protein which works together with the kinase portion of LRRK2 to regulate its activity. The biggest insight, however, has come out just recently, in August this year, I think. One of the biggest insights, one of the biggest problems as well, in a sense. It explains the microtubule biology that I showed you earlier from Bill Dower's group. And basically, it's the cryo-EM structure at 3.5 Angstrom resolution of the functional domains, the ROCK, core, kinase, and WD40 domain, shown here of the molecule, and how that basically binds to microtubules. It's quite curious in that to resolve the structure, and from this rather unfolded kind of picture here to a much more high resolution, better fitted structure on microtubules, they noted, these investigators, Deniston and colleagues, noted that basically the conformation, the shape of the kinase portion of the molecule is critically important to its binding. Basically, the mutations in LRRK2, R1441C, G, I2020T, and Y1699C, all preferentially keep the kinase domain in a kind of closed conformation, which allows its binding, its preferential binding to microtubules. And that's quite interesting in a way, because that binding actually inhibits both kinesin and dynein transport up and down microtubule rails, and can impair trafficking. Different types of inhibitors, kinase inhibitors, depending on whether they directly block the pocket, such as LRRK2 in 1 or MLI2, and keep the DFG, this is the G2019S that's mutated in LRRK2. You keep that in a hidden domain, or they keep it, or different mutations, or different inhibitors that basically bind on the opposite side of the molecule and basically also inhibit kinase activity, but allow the DFG domain to be outwards seen, have different effects. So though they all are just as effective at stopping LRRK2 kinase activity, they have much different effects on whether that molecule, that LRRK2 molecule, can bind microtubules. This is shown here very nicely. It's really a great read, I think. The question will be, though, of course, is what's the physiologic function of microtubule, of LRRK2's binding to microtubules and interfering with both kinesin anterior grade and retrograde dynein trafficking? Is there a physiological role for that? And is that intimately associated and linked with the sort of shorter term, if you like, trafficking, actin-based trafficking within synapses and within axonal terminals? I think they're probably going to be mutually related, and I think this is going to be a really critical question to answer for the context of LRRK2 inhibitor development. The last thing I'm just going to mention is, to move this forward and to try and understand this, we really need to have better models, and we're still in that phase where we're looking at modeling of kinase inhibitors and how they may provide benefit or provide risk to patients. So it's very important we get this right, because individuals who are LRRK2 G2090 carriers won't want to take a drug potentially for the rest of their lives unless we can know for sure, absolutely certain for sure, that there won't be side effects from the long-term use of such a drug. One of the things that we found that's quite insightful is another mutation, just to point out, VPS35 in families with Parkinson's disease. Here's one from Tunisia as well. It's a completely different system. It's a completely different mutation in a different gene, in vacuolar protein sorting, and I won't get into the biology of it. But the curious thing about it is that this mutation causes an increase in LRRK2 phosphorylation and causes an increase in Rab protein phosphorylation as well, causes LRRK2 activation. And I think it's an interesting way of trying to understand what LRRK2 biology is doing in an orthogonal model. One of the things we've looked at in this context, we made a VPS35D620N mouse, and we see big differences in dopaminergic function within the animals. We see major increases in VMAT2 and decreases in the dopamine transporter with time in those mice, and quite reproducibly, and this is just a knock-in mouse. And the beautiful thing is that we can completely rescue all of the phenotypes in these animals that are induced by VPS35 mutation with the application of LRRK2 kinase inhibitors. And this is an example here of basically rescuing the dopaminergic release that we see. This is stimulated release with stimulating brain slices with 2-minute intervals between stimulations. And you can see that in the mutant animals, the D620N mutant mice, you can see this increase in dopamine release, which can be rescued back to wild type with the application, in this case, of MLI2, Merck's kinase, LRRK2 kinase inhibitor. And you can see the effects on LRRK2 kinase, decreasing kinase phosphorylation levels. And you can also see the rescue in terms of DAT and VMAT2, DAT being normalized and VMAT2 levels being normalized as well. And even though this application was only done over 1 week. There are a number of more modifiers coming through for LRRK2. This is, again, unpublished data, but from the LRRK2 MJFF consortium. You can see here 768 participants in this group. Actually, it's 1,879 LRRK2 G2019S. Was it me? No, sorry, 1,810 G2019S heterozygotes in total in the study, but looking for modifiers of age of onset and penetrance. And this is unpublished, but just to give you a bit of a tease, there is going to be coming out fairly soon, I hope, in Annals of Neurology. But there are also more genes, more proteins that are involved, and I think they're intimately associated with both LRRK2 and trafficking on microtubules, as well as endosomal sorting, vesicular sorting. And this particular protein that's highlighted here is coronin 1C, which is involved in ER endosome tethering and cleavage to actually generate vesicles from endosomal tubules. It's getting deep into the biology, but what I'm trying to point out is that, you know, the more you study something and the more that we look at it, it's not so much that it becomes easier to understand, it just becomes a far more complicated beast in a sense. This is a picture just showing all of the loci now that have been implicated from genome-wide association. This is from Andy Singleton and the International Parkinson's Disease Genetics Consortium. More than 92 loci, more than 187 genes, and these are just genes with very modest effects on Parkinson's disease, which for the most part isn't a disease that's considered that heritable. Nevertheless, through these types of studies, I think we're going to find basically, and we are finding, not just major genetic insights as to why people have become affected, but also the ability to be able to model those particular mutations, those particular proteins in cells and in mice, as well as in man, and the ability to think about very strategic precision therapy approaches, such as LRRK2 kinase inhibitors, and getting the right type of inhibitors for the right type of patients for appropriate clinical trials to actually slow down, if not prevent, Parkinson's disease. So thanks very much for your attention. I think I better stop there.